It's such a pleasure to be back in Toronto and to have the opportunity to see so many friends and, in particular to be able to relay all the exciting news that's being made on so many different fronts in Waldenstrom. I do want to acknowledge a couple of people here. I'm sure you're going to have the opportunity to see them. Dr. Jorge Castillo, who is the clinical director of the Waldenstrom's Center at Dana-Farber, is here. You'll be hearing from Jorge later on. We have also Dr. Carrasco. Ruben has been working tirelessly on the transgenic mouse model with MID88 for Waldenstrom's. You'll be hearing all the exciting news on the grant that's been funded and will be funded, in support of his work by the WMCF. We also have Chris Patterson here, who's the administrative director of the Bing Center. And Chris is also responsible for running the international workshops on Waldenstrom's macroglobulinemia. And of course, all the progress that you'll be hearing about today would not have been possible unless we all came together as a community to make it happen. And for this, I want to acknowledge the invaluable support of the IWMF, the WMCF, as well as the Leukemia and Lymphoma Society, both in the US as well as here in Canada. It has been really a global partnership that has made all these advances possible. In this upcoming Tuesday, actually a very special thing will happen. We have partnered with the National Institute of Health in Washington, and the focus there is to try to identify what the predisposition is. Why is it that people get Waldenstrom? Let alone the MID88 story. The MID88 story tells you what drives the cells to grow and survive, but why is it that some people are predisposed? You just saw the wonderful presentation by Dr. Mattis, which I think is really second to none, Jeff. That soup to nuts that you gave us was absolutely stellar. But you also made it very clear that people can be predisposed. There are families out there where you see multiple generations of Waldenstrom. So the question is why? Why is it that you see this? So on Tuesday, we will be delivering the DNA, the genomes of 1,000 Waldenstrom patients to the NIH in this collaboration to try to find what that genetic marker is. And we already have an idea of what that genetic marker is, but this will become a very nice confirmatory study. But, thinking back to Arnie Smokler, who was just trying to find somebody else like himself, with Waldenstrom, here we are now, with, over 1,000 patients, of DNA being able to put that, forward. And that's really just the beginning. Waldenstrom's used to be this orphan. And now Waldenstrom's has become the paradigm for all these other diseases. They look to us and they say, this is remarkable that we can actually, sequence the DNA, discover mutations, model them in the labs, through various tedious work, which I'll show you some of today, be able to find the targets, make drugs against those targets, run clinical trials, and then get, approvals from regulatory agencies. In the US, the FDA, in Europe, the EMA, here, Health Canada, and many, many other countries. That's a home run. That's a home run. And Waldenstrom's is the paradigm now for all these other diseases to look at and try to model themselves as well. So I want to say, first of all, congrats to all of you for making all this possible, because it's really that energy that drives all the investigators, here in this room and around the globe that continue to make all this progress. And we had in New York, the meeting that Betty was alluding to, we had the 10th International Workshop on Waldenstrom's. 350 investigators, representing over 30 countries were there. And our closing ceremonies, were held in the United Nations building. And we had the opportunity to honor we actually had the opportunity to honor 4 humanitarians with the Peter Bing Humanitarian Award that was given there. And 2 of them are here in this room today. And one is Carl Harrington, the president of the IWMF, and the other is Chris Patterson, the center director at the Bing Center. So let's give them a round of applause. But as we all know, there's many more humanitarians that are making all this progress possible. And I want to acknowledge in particular the board of the WMCF, because you've always been there for us and others, You have tirelessly worked on behalf of Canadian patients, but you've also fundraised like crazy to make, many of the projects which I'll talk about today possible. So, with that in mind, it's time to talk about Waldenstrom's. And I always like showing this picture. Jeff already showed it to you, because when Jan Waldenstrom first described this disease, a lot of people didn't believe him. It took 15 years before there was a confirmatory paper. And his son would say, told me one day, when my dad put that manuscript in the mailbox, he said, wait till you see how much trouble this gets me into. Because he was describing a new, unique disease. And everyone else said, well, it's just myeloma and you've just got it wrong. And it took many years. I mean, this paper was actually published in 1944. And it wasn't until 2002 when we all met as experts in Athens, Greece, and declared this really a legitimate disease, that it wasn't really a clinical syndrome, but it really represented a unique disease with its own pathology. And it would still be, many years later, in 2011, that the discovery of the MYD88 mutation would come along, something that's found in 95% of all Waldenstrom's patients, to finally connect the dots, that there was, a genetic, problem, unique pathology, and also, a clinical syndrome. And so that's how long sometimes, it takes before people acknowledge that you were spot on. Now, this was a very nice slide, and I'm grateful to Jeff for showing this to you, because it really describes all the things, all the moving parts of Waldenstrom's, how patients can present with many different ways. I don't want to go over every detail, but suffice to say that you can have clinic, as Jorge and Jeff and Christine here and others do, and you may not see the same presentation of a patient on the same day. We see in Boston now about 1,200 Waldenstrom patients per year, and you see a lot of patients on any given day, and it's quite possible they're all going to present differently. But it's important to know this, because sometimes the way we treat patients differs depending on, whether they're coming in with a problem like Bing-Neal syndrome, when the disease is in the brain, or they're presenting with some manifestation of their IgM. So that's why it's important to not think that we have to treat everybody the same exact way. Now, for the most part, we've relied on rituximab. I'm going to let Dr. Castillo go over the details of this approach, and Christine is going to also talk about the way Canadians are treated with these drugs. But I think that the take-home message here is that usually combination therapy with Rituxan has gotten us to our best responses, our deepest responses, but that depth of response is so critical to how long we can keep patients, out of harm's way. But with these drugs also comes many side effects. And I think it's important to acknowledge these side effects, because if we're going to, do better for patients, we have to also make, our drug therapies more safer. And with Rituxan alone, we sometimes see this IgM flare, which can put people into trouble. But you can also see that it compromises sometimes the immune system. With fludarabine and bendamustine that we rely, a lot on, we can also see secondary malignancies. And of course, with Velcade or bortezomib, you can also see neuropathy. So these are all the things that we've been working very hard to overcome by looking at newer agents. Now, Jeff was very kind to go over the mutations for us, but I think this really was a pivotal moment, the discovery of the MYD88 mutation, because up to that point, we really didn't know what caused Waldenstrom's. People would come in, we'd tell them all about the research that was being done, but we really had no credible, target to be able to look to in order to be able to innovate in advanced therapy. And that's why this discovery, made possible by the funding from the IWMF as well as Dr. Peter Bing, at a time when whole genome sequencing was still a new art. There were very few people who knew how to, do the sequencing, and worse than that, there were even fewer people who knew how to read those sequences. Just think a moment, any cell in our body has 3 billion DNA molecules. And the mistake that causes Waldenstrom's is one of those DNA molecules out of 3 billion has flipped over or mutated. I mean, that's literally looking for a needle in a haystack, and it explains why despite so much effort by people prior to this, they couldn't really identify what was causative. Now, one of the things that Jeff very nicely alluded to is testing for MYD88. This can be frustrating. Even though most patients with Waldenstrom's have the mutation, the way you go about testing for this can differ. And sometimes you'll see that some centers will painstakingly pull out the Waldenstrom cells, they'll isolate them and then sequence them, and that results in a better detection rate. But that's not always possible in clinical laboratories. I mean, they have to get lots of volume out because they're testing for so many different things on so many days. So what you'll see in a lot of clinical laboratories is that they'll take the DNA from all the cells in the bone marrow. Sometimes they'll use peripheral blood, and then they'll sequence. And the platforms that they sequence on may not be as sensitive. And what this slide is supposed to remind me to tell you is that some of these platforms, you may miss up to 30% of patients who actually have the mutation, but because of the fact that they don't select out the B cells, the Waldenstrom cells, and use a different scaffold for doing their testing, it's quite possible that up to 30% of patients may be told that they have no MYD88 mutation. And this is actually a study from our own institution. We actually, at the same time, took almost 800 patients, and we ended up pulling out their Waldenstrom cells and sequenced them with a very, very highly sensitive PCR technology. And then the same bone marrow we sent to the clinical laboratory, and 30% of the patients actually were told that they didn't have the MYD88 mutation from the clinical lab, but they were identified in our laboratory. And this has been one of the reasons why at the recent IWMF Leukemia Lymphoma Society roadmap meeting, we identified the absolute need to come up with better testing ways so that we don't make these kind of mistakes, because they're so critical in the way we're going to be able to target patients, even now, with drugs like Ibrutinib. So you're probably all dying to know, what does MYD88 do, right? Yes, yeah. You see enthusiasm out there? Well, it's a very interesting molecule. It's really part of the way you and I are meant to respond to infection. And so right at the top here, and actually first before I do that, I just want to acknowledge this investigator in our laboratory. There's my pointer. Can you see the pointer? It's time to advance. Here's Dr. Guang Yang. Actually, I'm told if I do this, it's supposed to get, well, maybe not. He really has put his heart and soul into identifying all the pathways that MYD88 is responsible for. But look up here, at the top here, you see the toll receptors. These are the sensors that are looking for infection on our immune cells. And so normally when a bug comes across, touches this receptor, the receptor then gets all excited and it triggers that MYD88 molecule. And MYD88 is supposed to then start all this programming to make the cell grow and survive and expand so that it can fight the infection. And as part of the apparatus, it actually brings in BTK, Bruton's tyrosine kinase. That is what Ibrutinib targets. It's very essential to the way this signaling pathway works. And the reason why this BTK molecule may be particularly important here is because there are breaks that are normally supposed to stop MYD88 from signaling, which patients with Waldenstrom's lose. This is actually a very interesting study that we published last year. And up to 60% of patients with Waldenstrom will not only have the MYD88 mutation, but they'll also be missing a chunk of their chromosome 6. And in that chunk in chromosome 6, there are all kinds of regulatory genes, including the inhibitor of BTK. The actual molecule that's supposed to block BTK itself goes. And you also see other molecules that are very important in that pathway. And so you not only have a problem where the cell's been turned on with the MYD88 mutation, you're also missing the brake that's supposed to control it. And that's why we're very excited about Dr. Carrasco's work, because in the original work that he showed us that if you put simply the mutation of MYD88 in a mouse, you don't really get Waldenstrom's. You need something else to happen, and that something else is what he's looking at right now, and he'll be telling you all about his exciting work, but it involves really this particular study, which is taking out the brakes here. And I think it's really important to keep that in mind, because it helps us understand how Waldenstrom's evolves. Now, the MYD88 story is getting more and more complicated. And I'm not going to say it all here today. The rest of that work I'm going to present when we meet in Philadelphia, because we have to have something there to be able to offer as groundbreaking as well. So, right? Carl, we need Canadians at the forum, don't we? Yes, that's our plug. So this is going to get more and more interesting, but I'm going to tell you, MYD88 is like a giant octopus. It has the ability to put its tentacles on so many switches and turn them on so that the cell can grow and survive. And I want to show you one of these switches here. You see that molecule HCK that I'm showing you there? That was discovered by Dr. Yang, and what he found was that this is actually turned on by the mutated MYD88. And it's actually the molecule that actually turns on the BTK, allowing it to then join, the MYD88 complex. Again, that's what we target when we use ibrutinib. But it also turns on those other pathways, Irk and AKT. Now, most of you probably don't know what they mean, but they are very powerful growth and survival pathways. So again, it's the octopus, putting itself, on all these growth and survival switches and turning them on. And so you're going to see here, from what I'm getting at is if you just merely use ibrutinib, you may shut down that main pathway, but look at the other pathways that still can, remain on. And that's why we don't see complete remissions. You don't see complete remissions with ibrutinib. In fact, with many of the drugs that we use, we never get to complete remissions. Again, one of the, questions that came up at the roadmap, why isn't it we don't get more complete remissions? Because if we're going to dream about curing this disease, we have to start by getting more complete remissions, right? And so it's important in that context to then know about all these signaling pathways and to know what we need to work on. And we are working on it. In fact, we have made very potent HCK inhibitors with the help of the IWMF, and we're going to be talking about those inhibitors and all the exciting work with them at the forum. Okay, and so the other mutation, CXCR4, we have to give a lot of credit to Zach Hunter, who's shown here, because he took the whole genome sequencing data that we had used originally to identify the MYD88 mutation, and he's a computational scientist. He's also a great biologist, but he actually went back and rewrote all the programs, and made lots of contributions to the way we understand whole genome sequencing. And he comes back one day and he goes, you know what, I'm seeing all these mutations popping up in CXCR4 in patients with Waldenstrom. And with more advanced technology, we were ultimately able to show that up to 43% of newly diagnosed patients actually have this mutation. Now you may ask, well, what does this mutation do, right? Are you asking? Are you all on board here? It's actually a cool mutation. And it's unique to Waldenstrom. So far as we know, even though we've studied this across many other diseases, we see it here and there, maybe in a small handful of other diseases like marginal zone, maybe 5%. And that's a close cousin of Waldenstrom, so you always wonder, are we talking about the same disease? But this is actually relatively unique to Waldenstrom, which is why we need to know a lot about this. We're not going to get much help from our other colleagues because it's not on their radar screen. And the Leukemia and Lymphoma Society should be given huge credit because they're the ones who funded, this work. And so let's just talk about CXCR4. This is actually, again, like one of those antennas that sits on the cell. And it's sensing for that molecule that you see there, CXCL12. It's made by the bone marrow stroma. So this is one of the reasons why Waldenstrom cells love to go to the bone marrow, is because they get these little goodies that are secreted by the bone marrow itself. And so when CXCL12 comes in contact with the CXCR4, that antenna then signals. And the problem is that if you have the mutation, that antenna stays up. Normally, when it comes in contact with with its target like CXCL12, it's supposed to shut down. It's supposed to, once it senses, it's like a sensor, it's supposed to shut down and then, send off the initial report. But if you have the mutation, the antenna stays, on, and it continues to signal and signal and signal. And that signaling involves this AKT and Irk pathways that you see. And what's really critical to understand is these pathways can cause drug resistance. against Ibrutinib and many other drugs. And so that's why in this context, when you think about CXCR4, it's important to think about it as a gene that might have a particular impact on drug resistance. But we also learned through a lot of work that's been done in our group and others that it can also modify disease. If you have hyperviscosity syndrome or one of these people that has very high IgMs, most likely you have a CXCR4 mutation. In fact, we'll even guess that in clinic. When we have patients presenting, Dr. Castillo and I and the rest of the group will go line through line with every patient. You see somebody with an IgM of 10,000, even before we get to that person's genomic report, we already know in our minds that this is going to be somebody with CXCR4 mutation. And that's why disease presentation as well as drug resistance is the way to think about this mutation. So with all this data, we were very keen to study Ibrutinib. And with our ability to study Ibrutinib, we also wanted to see what the mutations, how they impacted response. This was a trial that we did that led to the approval of Ibrutinib in the US, Canada, and abroad. And Dr. Advani, who's up there, helped us tremendously. She did the first phase 1 study, so we already knew because of her work about the drug's activity in many diseases, and Dr. Palumba at Memorial Sloan also played a very, very critical role. And why I want to show you this slide, it's a very cold day in November in Washington, is because with the science that had been done, the genomics, the signaling work, as well as the early evidence of clinical activity, we got, which was remarkable, the first ever breakthrough in oncology for fast track approval. And it was a new category that had been created by the FDA to help accelerate drugs if there was really a lot of smartness attached to it. And the idea that Waldenstrom's was the beneficiary of this first fast track was absolutely remarkable. And we went back, this time 2 years later, or a little bit less than that, on a much sunnier, warmer day, as you can see, lots of smiles on everybody's faces, as opposed to that, terrifying look over here with the wind gusting at our backs. And we were able to secure the first drug approval ever for Waldenstrom's, that being for Ibrutinib. And so again, this represents what we all strive for, to be able to find, the genes, model them and be able to get drugs and ultimately get them approved for patients with Waldenstrom's. Now, what I'm going to do is I want to present to you an update of the trial itself. This is called the pivotal trial because it was pivotal in the approval of Ibrutinib. And when we published the results in the New England Journal of Medicine, we only had an average follow-up of about 19 months. With the follow-up now that I'll show you, we're up to 47 months. And this was actually a very sick population of patients. On average, patients had 2 prior therapies. Some had up to 9. And sometimes Castillo and I lose count how we ever get to 9. When you think about 9 therapies, how much reaching out do we need to do? And 40% of the patients were actually refractory to their last therapy, meaning they didn't respond to it. So I'll tell you, this is usually a population of patients where you start predicting, maybe lifespans of 1 to 2 years' time. So this is why we consider this a very sick population of patients. And yet what we saw with the drug was absolutely remarkable. The IgMs came down very quickly, the hemoglobins went up very fast, and you can see with the update, the best responses where the average IgM went from 3,500 down to 800, and the best hemoglobin response went from 10.5 to 14.2. These were, absolutely unheard of, endpoints at the time. And this is the update here to tell you that 91% of the patients responded, but the way patients responded differed depending on what their MYD88 and CXCR4 mutation status was. If you had no MYD88 mutation over here, you really didn't respond. You didn't get those deep responses. And if you did have a MYD88 mutation and also a CXCR4, you see the difference here. Those deep responses, particularly very good partial responses, which means at least 90% reduction disease burden, was much less if you had also a CXCR4 mutation. And it also took longer. It took 6 months on average to get there, as opposed to 2 months if you had no CXCR4 mutation. And so this, I think, very nicely made us all think about how we could use these mutations to help guide therapy forward. This is now the updated, what we call progression-free survival. Jeff explained to you, what that means. It's in essence the time that you don't really, that you're responding, to the therapy. At 5 years, 60% of the patients continue to respond, but within those categories, you see a different breakdown. If you have no MYD88 mutation, you progressed very fast. If you had both, the average time that you had with your disease kept in check was somewhere between 4 and 4.5 years. And if you had only the MYD88 mutation, you see they're going on strong. 75% still remain, with their disease under control at 5 years' time. So here too, we're seeing how the mutations play a role in predicting how patients do on ibrutinib. And this is also true in the frontline setting. My colleague Jorge Castillo has been steering this trial of ibrutinib by itself in treatment-naive patients. And here too, he showed us very nicely that the deep responses were very much impacted by having the CXCR4 mutation. But even look here, the time to get to that was 7 months versus 1.8 months. So you're seeing the impact that CXCR4 has. And why you really need to know this, and all of us need to know this, is because in the community, a lot of patients are put on ibrutinib and then 2 or 3 months later they're taken off because, it's not working. And if you knew in advance that you had the CXCR4 mutation, it can actually set expectation. And as I'll show you in just a moment, it may also give us a new way of tackling this particular problem. So let's talk about Bing-Neal syndrome, because this used to be one of the hardest things that we could deal with. We used to see 1 to 2 patients a year. Now we're seeing 4 to 5 a month. And this was very, very hard to treat. This is when the Waldenstrom's gets into the brain. And sometimes this happens very late in the course of the disease because as patients are living longer, this is becoming one of those sites where the disease can manifest itself. We used to have to admit patients and give them high-dose chemotherapy, and many times it didn't work and we created a lot of toxicity. But if you use ibrutinib, particularly at the higher dose at 560 milligrams per day, you can see responses in this Bing-Neal syndrome, and these can occur very fast, and we can also show simultaneously that we can get sustainable drug levels in these patients. We also talked about peripheral neuropathy. You heard Dr. Mattis say, what a bane of our existence, peripheral neuropathy is, because it's notoriously hard to treat unless you can stomp down that IgM and keep it away from those nerves. And this was actually part of the trial, the pivotal trial, where we had patients with peripheral neuropathy who had all progressed on rituximab. And despite that, after getting ibrutinib, 5 of them had an improvement in their neuropathy and 4 had stabilization of their neuropathy. So this represents, I think, one unique and new way to be able to treat patients who have IgM peripheral neuropathy. And we'll have more discussion because there are a lot of different types of peripheral neuropathy, and we'll talk about how we go about tackling each one of those. Now, there are other BTK inhibitors currently in play. It's just nice for you all to be familiarized with these drugs. There's acalabrutinib. This also, hits the BTK, protein. It's given twice a day as opposed to ibrutinib that's given once a day. And its level of activity as well as its side effect profile are almost similar to ibrutinib. The only exception being that you do get headaches during the first few months of being on acalabrutinib. This can be remedied with some caffeine, so when you get your prescription, if you get also a card for Starbucks, you'll be fine. So keep that particular profile in mind. Zanubrutinib is actually making incredible progress also in Waldenstrom's. They have shown that their drug, which also is given twice a day, is very effective in the treatment of Waldenstrom's macroglobulinemia with a very, very similar efficacy and side effect profile. And there is this large randomized trial that's currently in progress comparing ibrutinib to zanubrutinib, and hopefully this will read out later this year. So I think this is good from a number of different vantage points because it may also create the economic competition that's needed to make these drugs also more affordable. So if you'll permit me in the next few seconds, I just want to talk about ways we want to improve BTK inhibitors because, whenever you come up with a drug, the next thing you got to think about is how do we optimize it and make it work even better. And so there is this trial called the INNOVATE study, which actually wanted to see whether or not adding Rituxan to Ibrutinib would improve over Rituxan alone. And Dr. Castillo, as well as Dr. Buskey in Europe, had shown us that in the real community setting, most patients get Rituxan alone. And so it was a fair comparator in terms of going forward in this trial. The major lesson was, of course, that if you add Ibrutinib to Rituxan, you make the world better, by creating better responses in Waldenstrom patients. But what was also very interesting is that if you look at the responses, particularly in those patients, that had the CXCR4 mutation, there were fewer, particularly those deep responses. But what did happen in a very pleasant way was these responses happened faster, on average about 3 months, versus 7 months when you use Ibrutinib alone. So this may be the one niche where adding rituximab may be better, to the Ibrutinib. However, this is actually the sobering statistic, that the patients that had the CXCR4 mutation were also the ones who progressed earlier, much like I showed you in the work with Ibrutinib alone. And so this does tell us we do have a CXCR4 problem. And so what are we doing about that? Well, the good news is that CXCR4 itself can be targeted. So everybody in this room should get really excited about this. And there are a number of new drugs, including this monoclonal antibody called ulocupilumab. It took me forever to learn how to pronounce it, but you can actually give ulocupilumab with Ibrutinib in CXCR4 mutated patients, and that's what we're looking at in this trial that's also supported by the Leukemia and Lymphoma Society. The trial is still ongoing, but I'm going to tell you the data that we have back already makes us very excited. We're getting deep responses very fast, and I think this represents really a way forward. Now, to complement this, there's also another drug called the X4 drug, which is an oral inhibitor of CXCR4. So you don't even need to come in anymore to get your infusions as you do here. And that trial will be opening up later this year, combining Ibrutinib with the X4 drug in patients who have CXCR4 mutated disease. And again, home run, because whenever you discover mutations, it only is exciting when they're actionable, meaning you can do something against them. And so here we are now with both MYD88 and CXCR4 showing that these are actually actionable mutations. Now, I'm not going to take too much time to talk about this innovation because Dr. Castillo is going to talk about his trial, but Venetoclax is a drug that targets this very interesting molecule called BCL2. Now, all of you are hockey fans in this room, right? You are. Okay. Who isn't a hockey fan? You can't be. Well, I'm from Boston, yes. So I just want to remind you that. So you've got in hockey, you've got one team trying to score a goal, and you've got the other team with a goalie trying to prevent the goal from happening. And that's what BCL2 does. That molecule actually is a very pesky molecule. And so Dr. Castillo is trying to wipe out BCL2. And so making the hockey analogy, it's like trying to take the goalie out so you can start scoring. And when you put Ibrutinib into culture with Waldenstrom cells, you don't really see very much happening. You'll see a little bit of death early on, but not a lot. But if you combine it with Venetoclax, you can really stomp out those cells. And so the stomping out starts here with Venetoclax. This is also an oral inhibitor. And his data, which he'll show you in more detail, shows that it's a very, very active drug. And one of the ways that we're hoping to overcome, Ibrutinib resistance and get at those, complete remissions is to add Venetoclax to Ibrutinib. So I'm very excited about this trial that Jorge will be leading. Hopefully you'll be telling us a lot more later on this afternoon. But the ability to put these two very potent drugs together to treat Waldenstrom's is really what this study is all about. Now, I want to show you some of the most elegant science that has come out of our laboratory, and this has to do with Ibrutinib resistance. And we discovered, because of the work of Lian Zhu that's shown there in our group, we discovered all these mutations, in people who were becoming resistant after responding and doing well on Ibrutinib, for quite a while. And she found that there was a mutation, it's called the Cis-481 mutation. This is where Ibrutinib is supposed to bind to BTK itself. It's the anchor point. So if you have a mutation there, ibrutinib no longer can bind to BTK. And that's a bad thing because then the drug really can't be active. And so in our laboratory, this mother and son team, and you can see the resemblance there, have been working very, very, very hard to try to model this mutation in Waldenstrom cells, as well as cells from other diseases like Waldenstrom that have the MYD88 mutation. And what they discovered, and I'm not going to really hold you to this during the quiz, but you'll see this pathway called the Irk Pathway. We already talked about Irk a couple of times, but in all these cells, all those arrows there show you in all these cells, the Irk Pathway got turned on when you put in this BTK cis mutation. And so, here is the escape hatch by which these cells are able to outmaneuver ibrutinib, is through this Irk pathway. Now, there are things we can do about Irk, and again, I don't expect you to remember all these experiments, but just know it probably took years to get there, is that there are Irk inhibitors out there. We can actually kill these Waldenstrom cells that are trying to use this escape hatch for their survival in the presence of ibrutinib by adding on an Irk inhibitor. And for this reason, this paper, when it was published in Blood last year, really got a lot of attention. Blood is our top hematology journal. And we began to now have some interesting new insights into how these cells were becoming resistant to ibrutinib. And let me show you actually the part that I think is going to get all of us in this room kind of fired up. First of all, when you look for the mutation, the BTK cis mutation in ibrutinib resistant patients, it's usually a minority of the cells that have it. It's not most. And yet the patients are progressing on. And this has been a dilemma for us. And one of the ways that we theorized maybe these, the Waldenstroms was becoming resistant is by the secretion of these BTK mutated cells of proteins that actually could make the other cells, the other neighbors, resistant. And so that's exactly what we found. And this is, these are Waldenstrom cells that have been, fortified with the BTK mutation. These are the ones without. And you start seeing these little red areas here. That means that those cytokines, those proteins are now getting flamed up. They're actually being turned on, like interleukin 10. You see the red box here? That means that once we've introduced the BTK mutation to these cells are now secreting like crazy, this inflammatory cytokine. And we can actually shut it off with an Irk inhibitor, which is what it shows here in these boxes. All these inflammatory cytokines, you add an Irk inhibitor, and you see how they turn now blue? They're cooling down again. We're shutting that off. Now, one of the reasons why I think all of us in this, in this room can get really excited is because we can do all kinds of crazy modeling. And this represents to me one of the most exciting experiments that was done, where we did these co-culture experiments. We can actually put a membrane that can separate out two different populations of cells. So on one side, you can either put cells that have no BTK mutation, and the same on the other side, or you can put on the other side cells that actually have the BTK mutation, and then monitor and see what happens to the cells on the other side of the membrane. And what happened to the cells on the other side of the membrane in the presence of ibrutinib is they began to grow like crazy, because these cytokines, these proteins, were actually diffusing through the membrane and turning those cells into resistant cells. And so all this was being driven by communication from the mutated cell to the unmutated one, telling them to resist ibrutinib as well. And we know this because once we started using antibodies to shut down those molecules, we were able to reverse the resistance. And so if you can think about this analogy, that it only takes one bad apple to spoil the bushel, that's exactly what we're seeing here. And the real science of this is, how do we get rid of that bad apple? And so there are ways. In fact, there's an Irk inhibitor that has completed clinical trials that we're going to be using now to be able to silence this pathway forward. So instead of taking the patient off ibrutinib, which would be a mistake because the ibrutinib is actually able to handle the vast load of these Waldenstrom cells that don't have the mutation, you actually add on an Irk inhibitor and continue the therapy. And that way we can actually prolong the lifespan of people being put on these BTK inhibitors. And I think it's a really important thing to keep in mind, because as Dr. Castillo has shown us, if you take a patient off ibrutinib, even if you hold the ibrutinib, for a procedure, sometimes all hell can break loose. The IgMs go up, the hemoglobin goes down, they start getting fevers and night sweats and not feeling well. So that's what we don't want to do. We don't want people to have this, bad rebound. So therefore, you want to continue the ibrutinib and add an Irk inhibitor. So this is going to be a very exciting trial. It's not only for Waldenstrom's, but it'll also cover the other diseases that are using ibrutinib that also develop these mutations, and that includes CLL, marginal zone, and mantle cell lymphoma. Okay, I've got just a few minutes left here, but I just want to also remind everybody that it may not just be about MYD88 and CXCR4. There's a lot of very interesting work that was done early on in our laboratory that identified other mutations. I'm not going to get into the details of all these, but suffice to say, there could be other mutations out there that we already know about and suspect will be there that can also affect the disease and maybe provide new targets for therapy. And so this is a project which the WMCF has also played a very important role, as has also the IWMF. And it involved sequencing 300 patients with Waldenstrom, but sequencing them really from soup to nuts, trying to understand their DNA, trying to understand how the RNA is made, and then understanding what the modifiers for that are. So the good news is this entire project is getting close to completion. All these patients have been sequenced, and we're continuing now to finish up this part called the Epigenome. And that's where the WMCF has provided us with funding to actually look at this. And let me just show you this picture and leave you with this thought that we all have DNA, but think of it as the computer. But you also need software to drive the computer's programming. And that's where methylation, which is a form of epigenomic regulation, plays a big role. These methyl groups are like frosting that's put on genes, and if you're frosted up, that gene doesn't actually code. And so what you're giving us is the ability to actually look at these methylation profiling to try to explain why in patients with Waldenstrom's we have all these coding mistakes where the genes are not being turned on correctly. And so this has really been pivotal. We already have some very exciting preliminary data that we presented at the American Society of Hematology, alluding to how this mechanism is going to be very critical to understanding Waldenstrom's. You should also know that we are very excited also about studies that are trying to understand what happens to the DNA of these cells as patients get treated. Are we making things better or worse? This was identified last week as one of our roadmap priorities. So you should see that more genomic discovery will invariably be needed. And the last slide that I really want to show you here in terms of the genomics is, what if you don't have a MYD88 mutation? What happens in that population? Well, we know because of the study done at our institution as well as our colleagues at the Mayo, that those patients that don't have the MYD88 mutation are more prone to their disease transforming into an aggressive lymphoma. And here too, the genomics could help us, because when you look at the mutations that actually account for this, many of those mutations overlap with patients who have aggressive lymphoma. So we already know that they're already predisposed. But this is also a very valuable diagram to look at, because most of those mutations are actually below Bruton's tyrosine kinase. So this is why ibrutinib itself doesn't appear to have a role in the treatment of patients who truly have no MYD88 mutation. So I just want to conclude here by telling you that as we look at the future, our ability to actually leverage these genes can be very, very important. You can start thinking about, predominantly an ibrutinib-based therapy in patients that have only a MYD88 mutation, no CXCR4, and you can look at other therapies that can overlap, which we'll talk about during our break, for special conditions, like if you have somebody coming in with bulky disease or amyloid or peripheral neuropathy. You can think about the fact that if you have both mutations and you need a very quick response, like somebody presenting with severe hyperviscosity, that maybe ibrutinib is not the right answer because it may take a while. But if you don't need to get to an immediate response, maybe you need ibrutinib and Rituxan, or you start thinking about bortezomib or bendamustine. And then finally, this population worries us the most, those that don't have the MYD88 mutation, because here too, our ability to treat these patients still lacks, any cohesive targets. The same for patients who have been previously treated. I think the one nice thing to think about now is that we have Venetoclax, and I expect that this is going to be another very important drug, as Dr. Castillo is going to talk to you about, very shortly. And then finally, I just want to acknowledge all the members of the team back at the Bing Center who make all this progress possible. And my sincerest thanks to the WMCF as well as the IWMF and LLS for making all the funding that helps drive all this innovation possible. With that, I'll stop and thank you so much for your kind attention.